Okay, good afternoon. Good afternoon once again, and welcome to our today's virtual talk. My name is Richard Ngeno. I work here at Nature Kenya as Budding and Membership Officer, and I'm also the moderator of this talk. So welcome on board. And virtually we go through after an hour or so in this interactive and educative virtual talk. Today's talk has been organized by our MAMOL committee and will be presented by Winfridah Onyari. And the title for the talk today is Protecting Endangered Species Through the Lens of Science and Technology. Ladies and gentlemen, on Monday, 3rd of March, it was World Wildlife Day, and the theme was Wildlife Conservation Finance, that is investing in people and planet. This particular theme highlights the need for resource mobilization to protect wildlife. We are in that journey. And also, on Saturday, that will be on 8th of March, is International Women's Day. And we're really very proud for our MAMOL committee for bringing in on board today a lady. Also, we celebrate her, we celebrate all the women in conservation as well. And this year's theme is for all women and girls, rights, equality, empowerment. And actually this year's theme calls for action to unlock equal rights, power, and opportunities for all and inclusive future where no one is left behind. Welcome, ladies and gentlemen. It's my pleasure to introduce our esteemed speaker, Winfridah Onyari. She is a molecular analyst at the Wildlife Research and Training Institute, specializing in forensic and genetic analysis. Her expertise lies in conservation genetics, forensic science, and wildlife research. Currently, she is pursuing a PhD in animal genetics and breeding at the University of Nairobi, where her research focuses on population genetics and the effects of habitat change on cheetah populations in Kenya. Her work aims to understand cheetah population structure by analyzing genetic diversity, variability, relatedness, parentage, and the impact of habitat shifts on these endangered species. Her academic journey includes an MSc in medical microbiology from Kenyatta University, where she investigated the genetic diversity and antibiotic resistance of E. coli in wildlife, humans, and livestock in the Masai Mara ecosystem using a One Health approach. She also holds a PSc in medical laboratory science from Kenyatta University. With extensive experience in laboratory sciences, she began her career in hospital-based research in the year 2012 before transitioning to wildlife research in 2015, working with both the Kenya Wildlife Service and WRTI. She has received specialized training in conservation genetics, including leading the Congen Global 2025 course. Additionally, she has trained in wildlife crime management, focusing on combating wildlife trafficking, crime scene handling, and the legal responsibilities of forensic experts. Her dedication to wildlife conservation has been shaped by her academic background, professional fieldwork. She to contribute to science challenges in wildlife genetics and forensic analysis. As a brief of today's speaker, and I request all our participants that we remain muted so that we give the speaker our time to take us through the presentation. Also, if any question, any comment, the course of speaker's presentation, I welcome you to raise them in the chat box. Speaker will be able to go through all of them application. And also to let you know that this presentation is being recorded and we will be.
Wildlife DNA forensic is the use of wildlife crimes. It helps in fighting wildlife poaching, illegal wildlife trade, and habitat destruction. Hence, endangered species assist in the enhancement of conservation laws and aid in wildlife crime. The main reason scientific is used in solving wildlife crimes is because scientific evidence is a proof beyond reasonable doubt in the court of law. For a scientific evidence to be admissible in the court of law, it needs to follow the right evidence, documentation, and handling, and also to follow the chain of custody. Of my genetic research, I use DNA techniques to understand and protect key species in wildlife. I'm more specifically passionate, and this leads to my study in cheetah population genetics in Kenya. So why am I studying population genetics in Kenya? Globally, we know that climate and anthropogenic activities are threat to global diversity, meaning that effects of climate and man-made activities lead to biodiversity loss. And the cheetah as a species affected by these factors. The cheetah is the fastest land animal, meaning it can go up to 130 kilometers per hour. It's only the species in its genus Arachnops and the smallest of the big cats. Historically, this species used to be widely spread in Africa, Europe, North America, Asia, with an approximate population size of about 100,000 adult individuals. Today, it's basically spread in 9% of its historical global ranges, with a population of about 7,100 individuals. And these individuals in Africa, where we have the African species, and Iran, we have the only critically endangered species, Asian subspecies. And currently, cheetah have reintroduced to India, where they were extinct. The cheetah species is classified as the cause of decline, which is estimated at 2.1% globally, and also because of the reduction of habitats. This species has a unique evolutionary and social issue compared to all others. Kenya has a diverse ecosystem, including wildlife, of her socio-economic development because of ecotourism. This species force the species to either adapt or face extinction. And this extinction or adaptation and ecosystem disruption is most feared in large elusive carnivores with low population densities where the cheetah species falls into. Kenya is a stronghold for the East African cheetah population, which is estimated at 2,000 in East Africa, but Kenya holds this population at approximately 1,160, meaning Kenya is an icon of conservation to the East African cheetah population because it has the highest number of this population. The cheetah also is a wildlife animal, which is among the top that attract tourists to Kenya. Hence, the decline in cheetah sizes in Kenya will negatively affect the genetic resources of our country, ecotourism, revenue, and national development. Today, this species is distributed in Kenya in Laikipia Samburu ecosystem, Mara Serengeti, Machakos ecosystem, and Tsavo ecosystem. What are the challenges facing the cheetah? Cheetah has numerous threats to its biodiversity, but there's one special threat, which is historical population bottleneck. And when I say historical population bottleneck, I mean that this population went to a severe genetic bottleneck where their population was severely reduced in size and also in genetic loss, meaning that this species historically lost its genetic diversity at an early stage. And what is the consequences of loss of genetic diversity? The species is already vulnerable to environmental changes, including climate change. It's also vulnerable to diseases and inbreeding. Also vulnerable to diseases and inbreeding effects. The cause of inbreeding is because of the species having a very low genetic diversity, which affects the species adaptability and also can lead to low recruitment level of the cheetah cubs. Hence, population might not improve in its population size. Apart from the historical population bottleneck, the cheetah also faces the current threat to biodiversity, including habitat fragmentation and loss, human-wildlife conflicts, illegal trade and poaching, diseases, and interspecific competition. This current trend might lead to further reduction in cheetah genetic diversity to an incalculable extent, and hence limit their potential to adapt in the ever-changing environment. This because any threat to biodiversity leads to affect the biological fitness, genetic structure, and the conservation status of a species. Population genetics. Basically, population genetics is a science which studies the genetic health of a species by analyzing the genetic makeup of a population. When we do population genetics, why in cheetah as a species? Because cheetah face different threats and management challenges across its distribution range. Remember, cheetah species stays both inside protected areas and a large population, about 77%, live outside protected areas. The cheetah species is an elusive species with a low population density. Hence, by not examining its population trend and status, it means that this status might go unnoticed, especially outside protected areas. The conservation efforts of today demand that the cheetah species have up-to-date information on its population dynamics, habitat prevalence, and genetic status in order to help have species-specific conservation strategies. Hence, a comprehensive integration of ecological, genetic, and geospatial genetic data is needed in making critical decisions to ensure that this species is preserved for the future generation. The cheetah population genetic study will basically give us information on how this species is genetically diverse or inbred in their population. The second one, it will tell us on how this species interacts across the landscape. It also informs us about the cheetah-kin relationships and how they are mating in the wild. It will also identify whether there are hidden pockets of genetic variation and also help in creation of genetic population maps. At the same time, it's critical to study genetic population in cheetah as it will give us an overview to pin if there is inbreeding. And if it's there, then we can get ways on how to prevent further loss of genetic diversity by either doing translocation or other management strategies. The significance of cheetah population genetics help us to create species-specific cheetah conservation models. It also helps us protect and manage cheetah habitats, conflict resolution, that is human-wildlife conflict, especially in areas outside protected areas, and also preserve the genetic diversity and the natural resources in the cheetah ranges. It will also help us integrate and adopt novel models of demographic parameters, such as genetic diversity in management, actions, and policies, as well improve within population variation, as well as increase population connectivity via translocation. What is the anticipated output on cheetah population study? This is a national cheetah genetic survey, meaning it will give effective population size of the cheetah, their ranges, and also their distribution. The second thing, it will help us have an overall cheetah genetic health, including cheetah diversity, variation, connectivity, and inbreeding if it's there. It will also provide information about cheetah kinship and pedigree relationships, including mating patterns. It will provide more information about how cheetah move across the landscape and how these movements affect their social structure patterns. And finally, it will provide geospatial genetic maps to show which populations are inbred, which populations have different genetic diversities, and also create a good model for conservation in making concrete decisions which are based in scientific evidence. As they say, you can't do science alone. This project has partnered with many collaborators, including Action for Cheetah Kenya and Mara Meru Cheetah Project, among many other partners. The main mission of these institutions is to promote cheetah conservation through research, scientific research, community participation, and also education. The collaborators also act as a link between science and practice. This means that the conservation genetics from the conservation genetics of the cheetah, these institutions are key in transmitting this information to the field for better management. The goal of collaboration conservation is not to just save cheetahs today, but is to ensure that the future generations will still be able to witness the roaming cheetah in the wild. And in conclusion, I said that the future of conservation relies heavily on the fusion of science, technology, and field conservation to understand and manage the genetic health of the Kenyan cheetah for a better survival in a rapidly changing environment. Then a comprehensive analysis needs to be done about the cheetah genetics, their habitats, and how to solve and mitigate the threats involved in today's world. Together, Institute, Kenya Wildlife Service, and finally, Nature Kenya for providing this audience. Thank you.
Thank you very much, Winfridah, for the insightful presentation. So I welcome all the participants this is now the interactive session where we will have a one-on-one interactive session with our speaker who have taken us through the insightful presentation. So I see some questions have already come through the chat box, and we'll go with one. The first one, as from it's Nick Marty saying thank you, and Simon Musila, what caused the species to have or lose its genetic diversity? And another question is 1,160 count of cheetahs in Kenya based on count by helicopters or transect walk counts? Maybe you can respond to that.
I can start with what caused genetic diversity.
Yeah, what caused the species? Yeah.
The species genetic diversity. This species underwent what we call historical genetic bottleneck, meaning the species initially, it had a wide genetic diversity with large population sizes. But this bottleneck led to the species having reduced population sizes. And when we have a reduced population size in a population, it means that the numbers already have been reduced and the diverse genetics which was there has been lost. There is genetic loss, meaning that the genetic pool of the initial species can never be the same as the current species when a species has undergone genetic bottleneck. So the current species we have today has a history of having a low genetic diversity due to the bottleneck.
Thank you. Yeah, there was the second part of the question. Thank you for the response. Is 1,160 count of cheetahs in Kenya based on the count by helicopters or transect walk counts?
The 1,160 population in Kenya is per the Wildlife Research and Training Institute's census report, which is always done using transects and helicopters and all field-based methods for census.
Okay, great, thank you. There's another question from Leo Gasoha. What are your thoughts about the translocation of cheetahs from Kenya to India?
Okay, let me go to the side because what I initially said, I said that the current population, we have Africa and Iranian population. And currently, cheetahs have been introduced to the India where they were extinct. The current population in India didn't come from Kenya. It came from South Africa to India, so that they were trying to see whether they can turn the cheetah in India to their natural habitats. This was introduction stock from South Africa to India.
Okay, thank you for the response. From Jospat, now a good work. Peter Kingori, great presentation. Winfridah, John Joini, nice presentation. There's a question from Grace Mora. What methods do you use in your analysis? Any preliminary results?
Okay, the overall cheetah genetic structure, we use DNA-based methods. This includes we target mitochondrial gene for haplotype diversity, and also we target the nuclear gene. This one we use microsatellites, which gives the overall genetic diversity of a population. So we are using basically microsatellites and mitochondrial genetic markers for population genetic studies. And preliminary that, so far, we have done sample collection from all cheetah ranges in Kenya, including skirts, blood, tissue, and hair samples. So the laboratory analysis is starting soon.
Okay, thank you for the response. See, there are more questions really coming in through the chat box. Ivy Mugo, thank you very much for the detailed presentation. And Antoinette Miunga, thanks for the informative talk. Can't wait to see the results of the study and timely restore the cheetah population. Sandy is asking, has this study already been done or you are still doing field or lab work? I think she's kind of mixed up because of the both expected outcome and conclusion in the presentation.
The study is ongoing. Yeah.
The study is ongoing.
Is ongoing, but already the study captures 3 methods. We are using ecological data and genetic data. The ecological data already is there with also population maps, which shows how this population has been affected through landscape use, land use, and also human activities, especially infrastructure. And this one, I can, like in the slides I'm sharing here, this is a cheetah at the Samburu area, and it was killed by a road kill, meaning that as we expand our infrastructure, and it's good for a country to expand its infrastructure, we need to check how is it affecting the wildlife movement corridors so that we don't have mortalities or cases for roadkill. Especially this cheetah species is much affected by infrastructure.
Yeah. Thank you. And what is Nicholas is asking, what is the root cause for the decline of cheetahs in Meru according to the Mara Meru project?
We are yet to ascertain that, but the ongoing study will give a conclusive answer on that.
Okay. Thank you. From Simon, have you collected any data? Which sample did you collect? And have you subjected to DNA genetic analysis? If so, what have you found out from your studies?
We have collected samples nationwide, including cat samples, blood, tissue, and hair samples, and the samples are in the pipeline of analysis. And maybe soon I present the results. Yeah.
Okay, there's another one from Leo. What have you found so far with regard to our cheetah genetics? What is your conservation target to help restore their genetic diversity?
My conservation target is that if I do cheetah genetics and I find that there is structures in the cheetah which needs to be assessed, like inbreeding, then we can look for ways in which we can prevent a fatal loss of this species which already has genetic loss by either implementing policies such as translocating individuals from one place to another to increase the genetic pool, and also interspecific competition in a way that if this cheetah species faces multiple interspecific competition is those competitions which come from other animals, such as the other large carnivores like the lion, predation of cheetah cubs, also cheetah mortality rates, which increase. So making sure this population doesn't survive to adulthood. Then management strategies can be included in these models to prevent that loss.
Okay, thank you for that. Most of the combo is asking, from your distribution map, are there no cheetah sightings in Nyanza and Western?
As far as I'm concerned, they have not been sighted, but we will do more investigations on that.
Okay, thank you. John Juin is assuming human genetic diversity is 100%. What percentage would cheetah's genetic diversity be?
Maybe, I can guess, maybe around 30, 40. Yeah.
Okay. John Musina, thank you, Winnie. Will the results of your genetic studies inform possibilities of captive breeding for restocking the wild in the future?
That's a political question. I don't know if the government allows. Yeah.
Okay, that's good.
But
Yeah, there
it
is
needs
one
a
for.
conclusive strategy.
Okay. Timothy Kime, and now that the species underwent bottleneck and we don't want to have such in future, will you recommend establishment of cheetah sanctuaries in the country?
Yes, I will recommend that if we experience more population decline in Kenya.
Okay, this one comment from Gadija, very insightful, and Josephat Nav, okay, just asking about room for collaboration. What was your end goal? Any room for collaboration in the project?
It's open. As more partners, the many, the better. Science is all about collaborations.
Okay, thank you. So I think if you won't mind, you will drop your email or other contact details so that maybe Josephat Nav can do a follow-up, and also any other participant who would like to do a follow-up on the same.
Yes.
Yeah, okay. There is a question from Rehema Liai. Awesome presentation, Winfridah. How can molecular technologies such as DNA sequencing and microsatellites be utilized to not only protect endangered species, but also improve genetic diversity and resilience in cheetah population?
The genetic methods can be used to improve cheetah conservation and also other species by first identifying what is the level of this genetic, what is the integrity of the gene pool. Once that information is known, then from there, if there are any negative effects on the diversity, then we can use that information to create species-specific strategies, including moving populations from one area to another to make starting stocks, or also making sure we move species which are rated. The cheetah species stay in social structures. So we can try to move dominant males from one place to another to make sure that we spread the diversity across the region.
Yes, thank you. Thank you for the response. And Timothy Kime would like to know more about the active studies of cheetahs in Meru National Park. Are you here to visit the park or what is happening?
In Meru National Park.
Yes, okay. He's saying he's not aware of active cheetah studies in Meru National Park. Is there any?
There is by the Mara Meru Cheetah Project, where they follow the cheetahs and also have the pedigree for the cheetahs, and also the same NGO is also doing studies in the Masai Mara ecosystem.
Okay, great. From Hokian Kasiangan, what's your prospect for the cheetah population considering the research already done, and how does it work for these species? Also, is there room for collaboration and teaching within the research team through internships and also volunteers?
Yes, there is a room for collaboration, as I said. The cheetah population size in Kenya is only known as per today by census, which is the approximate of 1,160. Through the genetics studies, we are going to get the effective population size per region, and at the same time, we are going to do population estimates using the samples we have. As you know, the cheetah is an elusive species, meaning that you can see it, but very rarely seen species. At the same time, this species stays outside protected areas. So meaning that its visibility is rare. So through the genetic study, using the samples, we can do estimates of the population and give the actual numbers in the wild.
Okay, thank you for that. There is one question from Noreen Mutoro. Great job, Winnie. Would translocation of cheetah species to other areas be advisable, given that there might be two different cheetah subspecies in the northern and southern parts of Kenya?
Basically, translocations, before we make any translocation decision, we need to check the genetics of that population. And moving animals needs thorough decision making, which involves many aspects, from moving from one region to another, climate conditions. At the same time, there are genetics. So I cannot advise moving population from the northern to the southern parts of Kenya because it might create hybridization. And we don't want to have hybrid population of cheetah because this one might also lead to genetic loss. But within populations, translocations can be done, those which have the same gene pool.
Good to know that. And, yeah, there's one more from Leo. What are some of your success stories from Kenya in combating wildlife trafficking based on DNA evidence?
Most of the convictions which have been received in the Kenyan courts today is because of DNA genetic evidence, because this is proved beyond reasonable doubt, which cannot be dismissed in the court of law. Yeah.
Okay. I see there's a comment from Nicholas. Maybe Nicholas, are you able to unmute and ask or give more elaboration on the comment, Nicholas? Nicholas, can you hear us?
Sorry, in another session as well, yeah. So, yeah, I was just wondering about the Mara Mirror project. But some years ago, I don't think I've been up to Mirror for quite some long time as well. And the cheetah population was declining quite fast and almost disappearing. That's my worrying there, what the root cause to that one is, and how it will be affected, mitigated.
Yeah, thank you, Nicholas, for raising that concern. And this population, the cheetah species, need to be taken seriously in conservation, since the populations might be going down and go unnoticed. The genetic study, together with the ecological study, is important in making sure that we track the population so that we don't end up having an extinct population in the wild. So this study is targeted in that aspect, making sure that we know what we have and then create conservation strategies which make sure that we don't lose the population any further.
Yeah, thank you. Thank you very much, Nicholas and Winfridah, for the responses. Okay, there is one from Leo. Are there any adaptability differences between the northern and southern subspecies, or is it because of some genetic barriers between the north and south?
Adaptability between the northern species of Kenya or the northern subspecies of cheetah? Maybe she can clarify.
Yeah, maybe I can try to clarify that. I think from one comment from Noreen there, she mentioned there is hopefully like subspecies between the southern and northern parts of Kenya, I guess. So I was wondering like whether there are any differences in adaptability between the northern and southern subspecies, or is the differences just because there is some sort of genetic barrier between the north and the south?
Yeah, the northern subspecies, which basically is in the Samburu Laikipia ecosystem and towards the northern of Kenya, is a species which we suspect might be having another subspecies in, because this population is towards Somalia. And in Somalia, we have another subspecies. In Kenya, we have Acnox tubatus. But in Somalia, there is Acnox tubatus somoniaring, and we are suspecting maybe this species might be interbreeding in between. So we want to check this population of the northern, which species is it? And if there is any hybrid population in that species. Same as the southern subspecies. And this one, I mean the Masai Mara, whereby we have also the Tanzania species coming in and out of the Mara due to the transboundary in the Masai Mara ecosystem. So these results, we will get this answer once we know which subspecies in which place. Yeah.
Thank you. Thank you for the addition. There is a question from Kibet Kikoske. Do you have any cheetah project with NRT or Mbala Research Center currently?
The NRT or Mara Research Center, to me, I don't have, but my partners are widely partnered. They have many collaborators, and that's why I created the window that there might be more collaborators in this project. So I'm sure there might be, or there might not. So I don't know for now. Yeah.
Okay. It's also dropping in another one. Has there been any melanistic cheetah reported?
No, I have no idea of any being reported.
Okay, thank you. Ladies and gentlemen, thank you very much for your questions, and to our speaker, thank you very much for the responses. All those questions that we were handling, they came in directly through the chat box that has really been very, very engaging and very interactive and also very educative. So maybe we will still take some few more questions before we bring the session to an end. So I will give this opportunity to those who would like to directly ask their questions. If you can raise your hand, and then we'll give you an opportunity to unmute and ask your question directly. So it's open to anyone. Okay, maybe someone is preparing. Noreen Mutoro is responding to Kibet Kikoske. Yes, in the Salama Athi Kapiti area, there was a white melanistic cheetah called Pepo. Thank you very much, Noreen, for the addition. Okay, so anyone still with a question, if you can just unmute and ask the question. Or better still, you can drop it in the chat box. Samuel Kuna is asking if there's a photo of people. That's interesting. So Noreen, do you have any photos?
Can you hear me?
Yes, we can hear you. Go ahead.
Yeah. Just give me a minute. I check online. Yeah, but it was, I think this was already back in 2012. It was quite well known in that area. So I want to see on the internet if it's, yeah, if it was also documented and uploaded somewhere in the internet. But there was that was at least one of the melanistic cheetahs I heard of in Kenya. And I think there might be a possibility that it had a cub which was also melanistic. But let me just check on the internet and I can share.
Okay. Thank you. That will really be great. If you can get them before the end of the session, we will really appreciate. And also just to let our participants know that our today's speaker has already shared her contacts through the chat box. You can always feel free to pick them up from there for any follow-up questions or any follow-up information that you may need from her. And Grace Mora, is there a reason why you chose microsatellite markers over NGS methods? Winfridah.
Of NGS methods, next generation sequencing, yeah, there is microsatellite markers, broad markers, which can give population genetics study data, and they're cost effective. But next generation sequencing method is a broad method, which is a novel method in conservation genetics, but it's expensive method, yeah, to use in genetics. So it needs more funders for that. On my case, I use microsite markers because of their surface. But given an opportunity, I can do next generation sequencing of the cheetah samples there.
Okay, thank you. Thank you for that. Yeah, I see there's a link that has been shared by Rehema about the rare spotless golden cheetah spotted in Athi Kapiti Conservancy of Kenya. So yeah, people can actually, exactly. There's also another link that has been shared by Noreen Mutoro. Here is a link with information about Pepo. That's good. So you can follow that link and yeah, for the great photos of the spotless cheetah. And Kibet is asking, is there a way we can have this recording? I missed the start of this session. Thank you very much, Kibet. You can always reach out and we will share with you the link to our recording. Karibu sana. So any other person with a question before we bring the session to an end? Maybe you can raise your hand and we give you an opportunity, or you can still drop one or two more questions in the chat box, and then we'll be able to take them up before the end, before we bring the session to an end. Okay. Do you plan to collect samples from mounted and skin specimens? That's a question from John Musina.
Currently, we have samples for skin specimen from north of Turkana, northern Kenya, Turkana area. So we will include them in the study. And Dr. Mushina, if you have more, you can add them to our database.
Okay, yeah, thank you very much. So maybe if Musina has more, you can always get in touch with the speaker. And there are the chances of crossbreeding between northern and southern? Are there chances of crossbreeding between northern and southern, and what are the shortcomings? That's a question from debate.
About this answer, I will do another presentation once I get the results so that it be a concrete answer. For now, I might be speculating about it. Yeah.
Okay, thank you.
Yeah.
Thank you for that. So, ladies and gentlemen, since there are no more questions from the participants, okay, there's one from Devi Gatanwa. How are you preparing to protect cheetah, like in Mara ecosystem, whereby it said that there was a poisonous grass, why by the cheetah might kill its prey and die from there? I can't really understand this question well. Okay, Gatanwa, are you able to unmute and maybe reframe your question so that the speaker might can pick it up?
Okay, I can just answer. In this
Okay,
study,
go ahead.
we are also, apart from the cheetah, we are also focusing on the habitats where cheetah live. And in this case, we are going to look at each habitat and its ecological factors. And he's asking more of the Masai Mara, the poisonous grass. So this need to be researched more and find the components. Why is it poisonous? And how is it poisonous to the cheetah? So that we don't have cheetah mortalities due to the environmental conditions. Yeah.
Okay, thank you for that. I think we'll pick first question as the last one for the session. Hi all, on the issue of bottleneck genetic diversity in cheetahs, is there a way to reverse this in natural ecosystem?
There's no a way to reverse this. This already happened. The current population already underwent genetic loss. The only thing we can do to make sure that this population thrives for the future generation is by reducing the impact of the current cheetah threats, the current population, the current threats to biodiversity, such as mitigating human wildlife impacts, avoiding illegal wildlife trade, as cheetah is one of the species which is affected by the pet trade. Also making sure that the environment is good for their viability and multiplication. This can all be done by making sure that we make sure that the environment in which this species is well preserved so that we preserve the species.
Thank you. Thank you for the response. Yeah, so I think, yeah, we've been able to go through all the questions and the comments that came in through the chat box. And thank you very much, everyone, for your time to join us for the whole of the session. Again, for the participants who joined in maybe late, I've also shared the speaker's contacts if you would like to do a follow-up with her. So as I told you initially before, this particular talk has been organized by our mammal committee. And I think it will not really not be very fair enough if we just close the session before allowing one of the representatives from the mammal committee to just maybe tell us who the mammal committee is, and also for the mammal enthusiasts, how can they be part and parcel of the team actually to walk the mammal conservation journey together. Ladies and gentlemen, allow me to welcome the chairperson of the Mammal Committee of Nature Kenya, Leo Kasoha, to just say something briefly about the Mammal Committee. Leo, if you can hear me, kindly go ahead.
Oh, thank you. First, I want to thank Winfridah for the amazing talk, great insights. Thank everyone for turning out for this talk. It's been quite interactive, so many questions and so much interest in learning about cheetah conservation. Thanks much for agreeing to give this presentation. Basically, we as the Mammal Committee, mostly I like to say we are just a group of people interested in mammals, knowing about mammals, studying mammals. So we're just a community that is interested, and we welcome you guys. If you are not a member of Nature Kenya, please join Nature Kenya and feel free to join the Mammal Committee, and we will be glad to have you to learn together about mammals and continue expanding the conservation, protection, and the wellness of mammals. So that's it about mammals, and I've seen a guy I know who does elephant stuff. Maybe one day he will come in and talk about elephant stuff. Thanks much.
Thank you very much, Leo, for your update about the Mammal Committee, and thank you also very much for organizing this particular talk and bringing on board the speaker. I've also shared the email that you can reach out to us on if you really want to know more about what we do as Nature Kenya, and also if you are interested in actually for you to join our membership. And also below the email is our website, which you can still go and have a look about more on what we do. So thank you very much, ladies and gentlemen, for your time. We really appreciate. So maybe before we bring down the session to the end, maybe Winfridah, any parting shot to the participants before we let you go?
Ladies and gentlemen, I take this opportunity to say thank you for joining hands in making sure that the endangered species in Kenya and globally is well protected. Thank you.
Thank you very much. And also see Sese, let's register in this conference and find more info on the Kenya's wildlife. He shared an event up there, so you can you're welcome to register for that conference. Yeah, it's already shared in there. So ladies and gentlemen, thank you very much. It has really been nice having you virtually. And till next month, when we'll be having such a session again, I wish you all the best and success in every endeavor that you are pursuing. Thank you very much, and have a lovely evening ahead. Asanteni sana. We can leave at our pleasure.
Thank you so much.
Thank you so much.
Thank you. Bye.
Bye. Thank you.
Thank you. Bye.
Hi, Richard.
Hey, Leo.
Oh, there are still guys in here.
How are you? Just a moment.
Yeah, Santi for helping us to do this.
Asante sana. Just a moment. clear up something.
For sure.
Leo.
Yes, Richard.
Aha, sasa ni sisi peke yetu sasa.
Sisi kwa sisi.
Sisi kwa sisi.
Sisi kwa sisi. Unanisikia?
Video imeenda. Nakusikia.
Oh.
Unakuona.
Oh, nice. I hope it went well. Everything went okay. I think I enjoyed there was so much interaction, so much questions, which is awesome.
Yeah, I really like actually. Yeah, the questions really came in, a lot of them, and then
The questions was
that one really shows, how people have already have understood the presentation.
Yeah.
Ukiona pale hakuna maswali ujue hakuna kitu watu walishika.
It was not so scientific.
Yeah, it was not so scientific.
Yeah, sure.
Thank you very much for taking up my caution. I was seeing a lot of science coming up.
You preempted wrongly.
No. I could see from the introduction na nikajua hapa mtajiongelesha wanasayansi wenyewe.
Yeah, I like venye ameweka too general just to present about cheetah stuff, why that and that and that without could dive into hizo details zinge confuse wase.
It was really very, very nice. It was good.
Sure.
Yeah.
So we
Nice.
look forward to another one.
Yeah, you tell me when. I'm ready. I have someone else, 2 people in waiting, pangolin and elephant.
Also ready.
So you tell me if
What's
you are
your
ready.
name?
Usianze kusema sasa ni mammals tu kila siku.
I'll always prompt you.
Yeah?
I will prompt you.
Sawa. Yeah, you let me know when there is an opening and we'll jump on it.
Yeah, I will let you know.
Sawa. Oh, thanks.
All right, thank you very much, and we look forward to more of this in the year.
And don't get
Sawa,
lost too
asante.
much.
I'm not.
Tusikusahau tena.
Mniite nakuja mnanitreat.
Alafu naanza kukualert mapema, next month in April.
Yeah.
So May kutakuwa na Mammal Council Isambu Conservancy. Just waiting for the date.
Roysambu, so Isambu ama?
Si Roysambu, sasa Roysambu utaenda kuhesabu lini? Wow.
The human beings are mammals.
soy some.
Okay.
Soy, yes.
Yeah. Ukitumiwa email unipe tuende tuone huko
Sawa.
venye kunakaa.
Hapana,
Sawa sawa.
is there a checklist ya mambo za museum?
By the way, do you have a
Check.
checklist of mammals ya museum or ama anywhere in Kenya?
Yeah, we just have hatuna ya specific areas, but we just have a big mammal checklist, mammals you can expect in Kenya. But within inside that whatever checklist, it tells you sort of the distribution where you can find them. But we don't have kama those little booklets zenye mkonazo za birds, like birds of Meru National Park, whatever. We don't have hizo specific ones. It's just a general big one.
Did you see what they were using in Kapiti during the counts? Those gill riggers. So maybe actually, yeah, as part of the assignment, we've been trying to improve their bird one. So if you feel as a committee, you can also leverage on that so that you can also structure the one for mammals. I think they will really be very much willing to support you guys on that. Because you see them, they are still learning. Wajua ni watu wa ng'ombe, si watu wa wanyama. And they are actually bringing the conservation aspect on board. They want to run the conservancy like independently like that. So so if you actually see in whatever checklist that they were using for the mammals, okay, kwa ile yenye walikuwa wanatumia for birds, it had a lot of errors, spelling mistakes on misidentifications. Pasina mapicha yenye wametumia hapo. So I don't know whether you had a look at the one for mammals, if they are correctly done. But that's again an assignment. That
We will try to look into it. Sisi, like, I didn't even look at that checklist because, I'm just like, I know that and that and that. So
now then they don't know.
So, but yeah.
Na hii issue ya introducing water, they call water buffaloes.
Tukule water buffaloes.
Introducing what?
Ziletwe.
Water buffaloes.
Hiyo tukule sini ng'ombe. Watuletee zenye tutafuga.
Umekuwa na debate about it. What is your take?
Yeah, I know, like, I think most guys wenye wako against it ni kuna tu wale people who just have a protection kind of mentality, protecting stuff, or others have introduced you and then it might have to manage your nini hizo like quotas, how many we can harvest and all that stuff. So I think if we had good laws in place to protect hizo stuff, then it would be easy to implement. But I think we still have issues with regard to law enforcement ya to make sure ukisema we are supposed to harvest this and that and use this and that, like how do we manage the whole thing? So I think hiyo ndio tu the challenge. Otherwise it's not a bad idea, it's something that can be done only if we had good laws and implementation ya hizo laws in place.
Sawa.
Yeah.
Thank you very much. And
okay.
Where are you in Nanyuki or where?
No, I'm not in Nanyuki. I'm in Western Kenya.
Where you don't have cheetahs?
Where I don't have what?
Cheetahs.
What? Cheetahs?
Cheetahs.
Kuna network mzuri ya Chukwesa. Na tunakuona tu very clearly.
We have lots of birds around, primates, so snakes. So you cannot have everything, but you can have at least something.
Sawa basi, anyway, Nairobi, pass by to say hi.
Yeah, only if you are there, because
You
the
was
last
here, well,
the other time nikiwa huko unahepa.
kwa ilikuwa tu this week.
Juzi.
Alikuwa ameenda
Yeah, I was in holiday.
alikuwa ameenda kule
Kule pwani. Sawa.
pwani.
Haya, sawa, asante, bye.
Sawa, poa. Yep, bye.
Show me your wisdom.